Swayam Prabha. Digital India. Educated India.
Welcome to the course on Genome Editing and Engineering module number 3 on recombination. Today we are going to learn about homologous and non-homologous recombination processes. From your previous classes, you have come to know that organisms are continuously exposed to a myriad of DNA damaging agents during its lifetime. You also now know that DNA damage are broadly of two types: the endogenous damage, which is caused by reactive oxygen species which are derived from metabolic byproducts, then replication errors, DNA base mismatches and topoisomerase DNA complexes. The second type of DNA damage is the exogenous damage caused by radiation like UV, X-ray, gamma, and hydrolysis, plant toxins and viruses. You know that if this DNA which is damaged is not repaired, it will impact health and also modulate certain disease states in various organisms. Luckily, there are robust DNA repair and damage bypass mechanisms which essentially protect the DNA by either removing or tolerating damage to ensure an overall survival of the organism. Certain genetic or metabolic diseases occur when these repair mechanisms fail for any reason, which is an exception rather than an unusual event. Whenever there is a DNA damage, the organisms respond to it at the cellular and molecular level by activating robust DNA damage response pathways or DDR pathways, which physically remove or repair the damage in a substrate dependent manner. There are at least 5 major known DNA repair pathways which are active throughout different stages of the cell cycle, allowing the cells to cope up with the infliction of DNA damage by various agents. Briefly, these are the base excision repair, nucleotide excision repair, mismatch repair, homologous recombination and non-homologous enjoining. A few other specific lesions are also removed by direct chemical reversal and interstrate cross-link repair. Double strand breaks are considered as the most toxic lesion. These DSBs are generated by various ways, which is already known to you: the endogenous stresses or the exogenous factors. Besides these, DSBs may also arise through errors in DNA replication or as normal intermediates during programmed cellular processes such as meiosis or VDJ recombination, which generates mature immunoglobulin or T cell receptor genes from the separate fragments of the germline genome. These DSBs can be programmed to trigger beneficial genomic rearrangement during meiotic differentiation or the establishment of the immune system. So, DSBs are kind of double-edged swords. They are harmful on the one hand, but we can also exploit them in certain cases. Whenever there is a DNA breakage, particularly DSB, what are the strategies the cell adopts to repair those DSBs? Without the repair of the DSBs, we have emphasized time and again that the survival of the cell will be at stake, and if it survives at all, then it may lead to certain disease states. So, for the healthy existence of an organism, the DNA is repaired whenever there is an occurrence of DSBs, and this falls mainly into the following types. The one is the homologous recombination. In this strategy, the cell will undertake the repair of the DSB strand break, whereby in one chromatid the repair process takes place using the sister chromatid as a template. So, this is kind of a template dependent repair. The repair of breaks by homologous recombination therefore is a high fidelity process, as it ensures that all the genetic information at the break site is retained due to the presence of the template or the sister chromatid during its repair. The other process by which DSBs are repaired are the non-homologous end joining method, which involves the simple rejoining of the broken DNA ends regardless of the DNA sequence. This mechanism is error prone, as small deletions may be introduced at the break site. There is a third form which is similar to the second form, and this is therefore known as the alternative form of NHEJ and is frequently abbreviated as ANSEJ, the A coming from the word alternative, or simply as AEJ. So, these are the three main DNA double strand break repair pathways. The first one is the homologous recombination, the second one is the NACJ, the third one is the AEJ or ANHEJ. So, these are various pathways through which the DSBs are repaired, and you can see in each pathway there are many protein molecules involved in every step. We will be discussing about the protein molecules that help us in repairing the DSBs following a certain pathway, whether it is homologous recombination or NACJ or ALT-EJ. Now, from this diagram you can see whenever there is a double strand break, the DNA is either repaired by homologous recombination or any of the other two pathways. Now, a DNA break is a DNA break. How does the cell identify or take decisions which pathway to follow for the repair of this particular break over here, whether it will go the HR pathway or the other two pathways. So, in this case the decision is taken by the involvement of certain players. For example, the DNA ends are bound by either mRNA or Ku78T heterodimer, as you can see in this picture. Binding and retention of mRNA will shift repair towards HR, and binding of the Ku shifts repair towards NHEJ. So, these are the critical players which decides which pathway the DNA repair will follow. And there is a short range resection by mRNA, long range resection of the DNA and RPA binding. The RPA is exchanged for RAD51, which facilitates strand invasion, DNA synthesis and repair, HR repair. Homologous recombination promotes pairing between identical or nearly identical DNA sequences and the subsequent exchange of genetic material between them. As the name homologous recombination suggests, there must be some kind of homology present between the two strands of DNA. That is why it involves nearly identical DNA sequences. Homologous recombination is termed as the guardian of genome integrity, as it acts to repair DNA damage. Homologous recombination is found to be involved in rescuing of replication forks that are stalled for various reasons, such as the missing factors or a particular difficulty upstream of the fork, such as supercoiling or intense traffic of proteins. Homologous recombination is a highly conserved process, whether it is in bacteria or in humans. It serves to repair double strand breaks or single stranded gaps in the DNA. For example, in higher organisms there is a protein called RAD51, and its structure and functional homologue of the bacterial strand exchange protein is the RAK protein in bacteria. HA is also a driving force for the evolution of multigene families. This is something very interesting. The HRA is contain HR is considered as a guardian of genome integrity. So, it guards the genome at all times, but it is also a driving force for the evolution of multigene families at the same time. So, there is some kind of relaxation and escape somewhere in between which is allowed. Without these, the diversity would not have happened. So, HR is a very interesting phenomena which not only guards the genome on the one side, but it also allows evolution to happen, particularly in multigene families at the same time. HR plays a major role in evolution and genome dynamics by changing gene copy numbers through deletions, duplications and amplification. Intrachromosomal recombination between ribosomal operands or between mobile elements scattered into the genome leads to deletion or tandem duplications of large regions within the genome up to several hundred kilobases. The emerging technical application of homologous recombination today constitutes the basis of targeted gene replacement for gene therapy as well as for the precise design of engineered organism. In our course, we will be using this technique or depending on this technique to a large extent. The basic model for homologous recombination was largely derived from genetic studies in fungi such as Ustilago and Saccharomyces. Studies with the bacterium E. coli have provided us crucial biochemical insights into the mechanism of homologous recombination. In E. coli, about 20 genes are found to be involved in homologous recombination. They produce specific proteins which carry out each of the key steps in the homologous recombination pathway. So, you can see in this diagram the various steps involved in homologous recombination as well as alternative recombination repair pathways. So, let us examine the events that occur whenever a DSB happens. So, once a double strand break occurs, one of the event that follows immediately is the resection. Most molecular models of homologous recombination describe the process in three key steps. The first one is the strand exchange, you can see over here, then the branch migration and finally the resolution. Strand exchange involves pairing of the broken DNA end with the homologous region of its sister chromatid, followed by strand invasion to form a DNA crossover or holiday junction. This process generates regions of heteroduplex DNA comprising DNA strands from different sister chromatids. So, what happens is resection. First, there is a 5 to 3 prime resection of the double strand break which produces DNA ends with 3 prime single stranded DNA tails. This is followed by strand invasion. The free 3 prime ends invade a homologous DNA duplex, forming a DNA crossover or holiday junction and act as a primer to initiate new DNA synthesis. This is followed by branch migration, where the holiday junction extends the region of heteroduplex away from the initial site of the crossover. During branch migration, the holiday junction is translocated along the DNA molecule, extending the region of heteroduplex away from the initial crossover site. In the last step, the holiday junction intermediates is resolved by cleavage of the junction to form separate duplex DNA molecules again. The holiday junction resolution is therefore a very important step in this entire process. This is resolved by endonucleolytic cleavage of either the cross strands or non-cross strands of the junction. Let us now discuss the molecular mechanisms involved in the repair of DSBs. You can see in this figure certain proteins called RAC-BCD and then RAC-A over here. We will discuss the role of these particular proteins in the repair mechanism one by one. DNA ends resulting from a double strand break is processed by a multifunctional enzyme complex called RAC-BCD. The RAC-BCD is a sequence regulated bipolar helicase nuclease that splits the duplex into its component strands and digests them until it encounters chi site, which is a recombinatorial hotspot. The nuclease activity is then attenuated and RAC-BCD loads RAC-A into the 3' tail of the DNA. So, these are the critical steps. One thing we have to remember that this RAC-BCD has two functions. One is it opens the DNA with its helicase activity. The next is its nuclease activity. The chi site is a short stretch of DNA in the genome of a bacterium near which homologous recombination is more likely to occur than on average across the genome. For this region, chi sites are also referred to as recombination hotspots. Chi sites serve as stimulators of DNA double strand break repair in bacteria, and its sequence is unique to each group of closely related organisms. In enteric bacteria like E. coli and Salmonella, the core sequence, for example, is 5' GCT GGT GG. In addition, it includes about 4 to 7 nucleotides to the 3' side of the core sequence, which play important role. Let us examine the primary structure of the RAC-BCD enzyme. The total number of amino acid in each polypeptide will be shown in the figure, which we are going to study in the next slide. This RAC-B protein, which is a component of the RAC-BCD enzyme, is a modular protein or modular subunit. The N-terminal domain contains 7 motifs, which is characteristics of SF1 helicases, and these are involved in opening up of the DNA double strand molecule. The C-terminal domain contains motifs characteristics of a diverse family of nucleases. The nuclease motif contain key catalytic aspartate and lysine residue. So, in this picture you can see the simplified diagram of the RAC-B protein, which contains around 1180 amino acid residues, and as already told to you, it is modular, having 2 modules, one having the helicase activity, the other having the nuclease activity. The N-terminal domain contains the helicase function, and it has 7 characteristic SF1 motifs. As you can see here, 1A, 1A, 2, 3, 4, 5 and 6, total 7 motifs. The nuclease activity lies in the C-terminal domain, and you can see here NUC motif, which marks the position of the nuclease motif, which contains key catalytic aspartate and lysine residues. The other 2 components of the RAC-BCD protein are RAC-C and RAC-D. So, in RAC-C there are around 1122 amino acid residues, and these contains the important region called as the chi recognition site. The RAC-BCD protein has around 608 amino acid residues, and it contains 7 conserved SF1 helicase motifs, as in the case of RAC-B, and you can see those in the yellow color strips in this diagram. So, we now know that RAC-B is a modular protein having helicase and nuclease activity, RAC-C is having the chi site, and RAC-D is again having the helicase activity. All these 3 join together to form the RAC-BCD protein complex, and in this you can clearly see that RAC-B and RAC-D, which contains the helicase activity, will be involved in opening the DNA double strands. This chi site is the place where due to which the stoppage of these molecule will happen, and a part of the rec B, the nucleus domain, you can see lying over here in this position. These are all schematic and not exact locations, but you can see in this particular protein complex the different functional domains with respect to one another. Now, what are the different functions of these different constituent proteins? The rec C recognizes the chi sequence, as already told to you. Once it recognizes the chi sequence, it signals rec D to stop, not to go further. Rec D will then stop and signals rec B to cleave the DNA. Rec B cleaves and continues unwinding DNA and loads the rec A molecule. Let us look into the various steps where rec BCD catalyzes DNA and processing resection. So, there are various steps laid over here 1, 2, 3, 4, 5, 6. We will discuss these steps one by one. Rec BCD binds tightly to a blunt or nearly blunt DNA end of a linear DNA duplex. The rec BCD couples the hydrolysis of ATP to DNA in the next step, and the DNA translocation and unwinding due to the helicase activity takes place. The single stranded DNA products are cleaved asymmetrically, with the degradation of the 3' terminated SS DNA tail being much more vigorous than the degradation of the complementary tail. In the next stage, the enzyme continues to translocate until it pauses as a correctly oriented chi sequence. At the chi sequence, the biochemical properties of the enzyme are altered dramatically. After chi recognition, rec BCD facilitates the loading of the rec A protein onto the 3' SS DNA. So, you can see the chi site here. So, once it arrives at the chi site, after that it will facilitate the loading of the rec A protein. After this, the enzyme continues to translocate, but the nucleus polarity is switched. The degradation of 3' SS DNA tail is attenuated, whereas the hydrolysis of the 5' SS DNA tail is upregulated. Rec BCD then repeatedly deposits rec A promoters, which act as nucleation points for filament growth primarily in the 5' to 3' direction. Following these, the rec BCD enzyme dissociates from the DNA. The product of the enzyme is a recombinogenic nucleoprotein complex of rec A protein bound to the 3' SS DNA tail with chi at its terminus. This product invades homologous duplex DNA to promote the recombinatorial repair of a DSB or to restart DNA replication. It has been found that the permanent inactivation of rec BCD enzyme by chi sites in duplex DNA occurs by the disassembly of the enzyme into its 3 constituent subunits. It is hypothesized that this inactivation occurs in 2 distinct steps. Number 1, upon entering a chi sequence, rec BCD enzyme undergoes its first change. It retains its ability to travel along the DNA and cut a hairpin DNA structure at the distal end of the DNA, but loses its ability to nick at subsequently encountered chi sites on the same DNA molecule. The second change, the disassembly of the enzyme into 3 inactive subunits, may occur either during continued unwinding beyond chi or upon reaching the end of the DNA molecule. Now, let us study a little bit about the rec A protein, which binds to the single stranded DNA and helps in the strand invasion. Rec A protein is about 350 amino acid residue long. Its sequence is highly conserved among eubacterial species. It is also found in the chloroplast of plants. Rake-like proteins are found in archaea and diverse eukaryotic organisms like fish and yeast, mouse or humans. The Rake protein of E. coli has around 353 amino acid residues. The Rake protein is involved in homologous recombination as already known to you, and it bypasses mutagenic DNA lesions by the SOS response. Rake protein catalyzes various reactions like the ATP driven homologous pairing and strand exchange of DNA molecules necessary for DNA recombinatorial repair. Then the hydrolysis of ATP in the presence of single stranded DNA molecules, and ATP dependent uptake of single stranded DNA by duplex DNA, and finally it catalyzes the ATP dependent hybridization of homologous single stranded DNAs. Let us now study about the various structural domains of Rake protein. You can see in this picture various domains which we have colored, and these colors represent certain domains which we will be discussing one by one. Rake is composed of around 350 amino acids and contains 3 major structural domains. There is a central core ATPase domain or CAT domain which extends from the 34th to the 269th amino acid residue, and here we are depicting these in the green color. Apart from this core ATPase domain, there are 2 other smaller domains, the N-terminal domain NTD and the C-terminal domain. So, we can divide this into 3 parts, the CAD, the NTD and the CTD. The NTD and CTD extends from the 1st to 33rd residues and from the 270th to the 352nd amino acids respectively, and shown here in the color red and yellow. Then there are various other important features in this particular protein. You can see certain flexible region. There are 2 flexible regions, then there are 2 important sites called site 1 and site 2. Then you have a dsDNA gateway. We will study about these various flexible regions as well as these MG2 binding sites and various site 1 and site 2 domains in our next slides. Let us first focus on the ATPase core domain. It includes different sites. Number 1 is the ATP binding site between the residue 66 to 73. Then there is site 1, as you can see over here, having the residues 157 to 216. This is a single strand binding, single strand DNA binding site. Site 2 is double strand DNA binding site. So, this particular molecule can bind to single stranded DNA and double stranded DNA at the same time, and for this it has specific sites, site 1 for ssDNA binding and site 2 for dsDNA binding. It also includes residues responsible for the ATP hydrolysis activity. You can see these in the residues around 240 to 250 here into here. This C-terminal domain can be divided into 2 domains, the dsDNA gateway domain between the residues 270 to 328 and the CTD tail between the residues 329 to 325, which modulates the protein activity. The Mg2 plus binding is coordinated by D144 and the CTD tail as shown in this picture. Now let us focus on the flexible regions. There are total 4 flexible regions of the protein, and these are depicted by these violet boxes. So these regions are the regions between residues 24 and 37 at the end of the N-terminal domain and the beginning of the core binding domain. Then the next flexible region is L1, 157 and 164 amino acid residue. The third one is the L2 or the loop 2, and this lies between 195 and 209 amino acid residues. The last one is the CTD, which lies between 270 and 352 amino acid residues. So we now know the various structural domains in a RAC protein, which binds to RAC BCD, which is being loaded into the single stranded DNA by RAC BCD, and then finally which drags the RAC single stranded DNA into a duplex DNA structure. How does it act? What are the mode of its activity? ssDNA and RAC-A, along with its cofactor ATP, form an active right-handed helical nucleoprotein filament with 6 RAC-A monomers per turn. These active nucleoprotein filament will search for and capture a homologous double-stranded DNA to produce a synaptic structure. Once a region of homology is found, the single-stranded DNA on the homologous chromosomes are exchanged, producing heteroduplex DNA structure. The nucleoprotein filament has hydrolytic activity, and this hydrolysis is carried out by the Kr into Kr hydrolysis motif, which contain lysine 248 and 250, which cooperates with glue 96 on the other monomer. The single stranded DNA binds to site 1 of the protein, forming a nucleoprotein filament. Then ATP, a RAC-A cofactor, binds to the ATP binding site and activates this filament. After that, RAC-A performs a homology search to find a homologous double strand DNA. In this process, double stranded DNA interacts first with the NTD and then with the CTD, by which it can move to site 2, where it will bind to double stranded DNA. If the bound double stranded DNA is homologous to the single stranded DNA, strand exchange is performed. If it is not, the double stranded DNA is released. So in many ways, this is some kind of a random interaction where it tries to do some matchmaking. If the matchmaking happens, the reaction will proceed forward. If the matchmaking fails due to the non-availability of complementary sequences, the double stranded DNA will be released and the system will keep on trying. Let us now move to the homologous recombination in eukaryotes. Most of our studies, discussions we had till now are based on prokaryotic research. The process is similar in eukaryotes, but there are certain players which are unique to the eukaryotic system. Here also resection takes place, and the double stranded breaks are recognized by MRE11, RAD50, NBS or MRE11 complex. The capture of DNAs by the mRNA complex will lead to the rapid activation of the ATM kinase, which phosphorylates diverse substrate participation in DNA damage response. BRCA1 and P53 binding protein 1, 53 BP, here you can see this, they have opposing roles and influences the choice of homologous recombination over non-homologous enjoining and potentially other mutagenic pathways of DSB repair. So, in the case of eukaryotic DSB repair, the involvement of BRCA and P53 are very important in the decision making whether it will go HR recombination pathway or other recombination repair pathway. Apart from number 1 initiating the resection, BRCA1 also helps in the loading of the RAD51. Let us discuss about the loading of the RAD51 on ssDNA. So, earlier we were discussing about the loading of the RAK on the single stranded DNA, and you already now know in details how that loading and strain invasion takes place as a result of that. So, here also there will be loading of the RAD51 on the single stranded DNA, and this complex will search for homology and helps in strand invasion. The 3' ssDNA stretch created by resection is coated with replication protein A, and it protects it from digestion. You can see here the involvement of the RPA proteins. The loading of RAD51 on the ssDNA is performed by the BRCA1 PAL-B2 complex. Okay. So, you have the BRCA2 PAL-B2 complex over here, sorry, not this one, here. So, this protein complex interacts with BRCA1 and catalyzes the replacement of RPA by RAD51 on the stretch of the 3' ssDNA, creating the RAD51 ssDNA presynaptic complex. The ssDNA RAD51 filament scans the genome to search for homology, and on success the filament invades the duplex homologous DNA and initiates strand exchange, creating a displacement loop. So, you can see here the events taking place sequentially. First, there is DSB recognition and resection, and here are the essential factors for this particular step, and these steps these are not essential in this stage. The second step is the strand invasion and D-loop stabilization, where BRCA1, BRCA2, PAL-B2, RAD51 and so many different players are critical for carrying the process forward, while these 3 molecules at the end are not essential factors, non-essential factors. How DNA synthesis happens once the strand invasion is successful, because it found out a homology sequence and the structure is now stabilized and DNA synthesis can take place. The DNA 3' invading strand will act like a primer. So, it will prime DNA synthesis through the recruitment of DNA and the copy of the invaded DNA molecule. Numerous polymerases are involved in this process, although delta polymerase has been proposed to play a primary role in this process. A protein complex HROB, MCM8, MCM9 and HELQ are proposed to have redundant helicase functions to promote DNA synthesis during this homologous recombination. Once the DNA synthesis starts and completed, the formation and resolution of the intermediates, HR intermediates will have to take place. The strand invasion and DNA synthesis lead to the formation of different intermediates whose processing leads to gene conversion either associated with crossover products or not. The invading strand can be disassembled, channeling DSB repair towards synthesis dependent strand annealing called SDSA. If stabilized, the D loop can lead to DSB repair by break induced repair or to the formation of double holiday junctions that can be either dissolved by the BLM top 3A RMI1 by 2 complex or resolved by the structure specific resolvase. This BLM is a RACQ family DNA helicase. It is mutated in Bloom syndrome and plays several roles, sometimes which are contradictory. For example, it has been shown to suppress crossovers in mitotic cells while repair mitotic DNA gaps via homologous recombination. BLM is involved in different steps of homologous recombination, including AND resection at HR initiation, D-loop rejection, and double Holliday junction resolution at the HR termination. At resection initiation, depending on the cell cycle phase that modifies its interacting partners, BLM favors the loading of 53 BP1 on the DSB in G1 phase, preventing the initiation of unscheduled resection, or in contrast favors resection in S phase when interacting with top 3. Besides these players, there are several other accessory proteins which are required in the process. For example, we need RAD54, which is a member of the SWI2 oblique SNF2 protein family, which are an ATP dependent chromatin remodelers. It interacts with RAD51, and in vitro studies have proposed that it functions as a RAD51 cofactor. RAD54 catalyzes the extension of joint molecules and stabilizes the D loop. A family of 6 proteins, RAD51B, C, D, XRCC2, 3 and RAD51AP1, known as the RAD51 paralogs, which are proteins that share sequence homology with RAD51 in a given species, have been identified in mammals. 2 distinct complexes have been identified, RAD51B, C, D XRCC2 and RAD51C XRCC3. RAD51 paralogs favor the recruitment of RAD51 to DNA damage sites and promotes the formation and stabilization of the RAD51 nucleoprotein filament. The RAD51 paralogs also influence gene conversion track length. The SWSAP1 protein, a non-canonical paralog of RAD51, forms the so-called SHU complex. When associated with SWS1, SHU interacts with the RAD51 and regulates its function. NHEJ. Let us discuss about the NHEJ pathway now. You already know how the homologous recombination pathway operates. Now we are going to discuss about the NHEJ pathway and the various players involved in it, Ku 70/80, then DNA PKCs and so on. The canonical non-homologous end joining pathway depends on Ku heterodimer and DNA PK catalytic subunit, which together form DNA PK holoenzyme. Ku is the first responder at DSBs and provides a docking site for DNA PKCs. Without the presence of Ku 70/80 in the breakage site, DNA PKCs cannot be recruited to it. Unlike mRNA, which can bind internally, Ku requires a free DNA and for binding and cannot associate with most block ends. Several nucleases including tyrosyl DNA, phosphodiesterase 1 and 2 and artemise can remove hairpins, damaged bases of protein DNA adducts. The DNA ends are processed by additional enzymes and rejoined by the LIG4, XRCC4 and XLF complex. So, you can see here the various players like mRNA, CTIP and Artemis, etc. Let us now compare the enzymes present in prokaryotes and eukaryotes which carries out the non-homologous end joining process. So, here you have the prokaryotes, and for the tooth pill protein you have the Ku protein which is around 32 40 kilodalton, and in eukaryotes you have the Ku 7080 in Saccharomyces cerevisiae, while in multicellular eukaryotes also you have the similar kind of molecule. The polymerase action or the carried out by the Pol domain of the Lig-D. Here it is carried out by Pol 4 in Saccharomyces cerevisiae, and in higher eukaryotes Pol mu and lambda are involved. The nuclease activity in prokaryotes, it's still not understood, although many reports have suggested certain candidate proteins. In eukaryotes, it is the RAD50 MRE11 XRS2 complex, while in multicellular organisms it is the Artemis DNA PKCs. The kinase phosphatase activity lies in the P domain of Lig-D in prokaryotes. In eukaryotes like Saccharomyces cerevisiae, it lies in TPP1 and others, and in multicellular organisms it is present in the P and K. The ligase activity in prokaryotes again lies in the Lig domain of Lig2. In eukaryotes like Saccharomyces, it lies in the NAJ1, LIF1 and DNLF4, while in multicellular organisms it lies in XLF, XSCC4, DNA ligase 4. So, from this comparison we can see the diversity of the protein molecules and players which are involved in prokaryotes, and also within eukaryotes, lower eukaryotes and higher eukaryotes have different proteins which carry out certain defined functions in the NSEJ pathway. The Ku is a critical player without which the NHEJ repair cannot begin. This is abundant and highly conserved DNA binding protein found in both eukaryotes and prokaryotes, and it plays essential role in the maintenance of the genome integrity. In eukaryotes, Ku is a heterodimer comprised of two subunits, Ku70 and Ku80, and it is best characterized for its central role as the initial DNA end binding factor in the classical non-homologous end joining pathway, the main DNA double strand break repair pathway in mammals. Ku binds the double stranded DNA ends with high affinity in a sequence independent manner through a central ring formed by the intervened strands of the Ku70 and Ku80 subunits. At the break, Ku directly and indirectly interacts with several CNHEJ factors and processing enzymes serving as the scaffold for the entire DNA repair complex. What are the general steps of non-homologous DNA end joining? It starts with the binding of Ku to the DNA ends at the DSB, and it improves the binding by nucleases, polymerases and ligase components thereupon. Flexibility in the loading of these enzymatic components, the option to load repeatedly and independently processing of the two DNA ends all permit mechanistic flexibility to the NSEJ process. This mechanistic flexibility is essential to permit NSEJ to handle a very diverse array of DSB end configurations and to join them. In addition to the overall mechanistic flexibility, each component exhibits enzymatic flexibility and multifunctionality. So, now you know about the homologous recombination and non-homologous recombination repair pathway. In addition to these, conventional NSEJ and homologous recombination repair, there is a third pathway of double strand break repair, which we call as ALT-EJ or ANH-EJ. It is functioning on simple end joining principles, but repairing DSB in a slower speed than conventional NSEJ. This repair pathway is considered to be an alternative form of the NSEJ, as already told to you, and is abbreviated as ANH-EJ. A NHEJ or B NHEJ. A NHEJ or AEJ is suppressed by C NHEJ. The conventional NHEJ will not allow the alternative NHEJ to operate, and it is also possibly suppressed by homologous recombination repair pathway. It is the least preferred pathway because of the suppression by the other two pathways, and it becomes active only when the standard repair processes fail. The CNHEJ and HRR fails to take care of the genome integrity. Under that circumstances only alternative NHEJ will come into action, and this repair may take place at the local level to the global level. For this reason, this is considered as a backup pathway. If conventional NHEJ and HRR fails, then alternative NHEJ comes into action as a backup mechanism. So, for this reason, it is also sometimes preferred to be called as BNHEJ by certain groups of people. So, several factors have been implicated in alternative NHEJ, and the functional diversity has led to the postulation that there are several sub pathways in this operation. It is not just one single pathway, and it engages prospectively at each double strand break on the basis of as yet undefined parameters in competition with other repair pathways. So, although it is the last one to operate when the first two fails, the entire landscape of its operation and mechanism is still largely unexplored. So, this AEJ will engage at DSBs, as already told to you, when either of the two pathways have somehow failed to act. Thus, at each DSB where AEJ engages, factors of either CNHEJ or HRR, particularly those involved in early steps, will be present when this alternative NHEJ takes DSB processing over. So, CNHEJ and HRR tried to repair the DNA, so those players are present, but they fail to do it. Then this process is taken over by alternative pathway. So, in the alternative pathway, we will always find the presence of the initial players involved in the earlier two processes. Also, it is possible and even likely that these factors have already operated at the DNA ends. So, resection has taken place and it has carried out one or more of the initial steps of the CNHEJ and HRR, which of course alters the state of the substrate presented to the alternative NHEJ. Furthermore, the presence of the CNHEJ and HRR factors at the DNA ends may either facilitate or compromise the operation of A alternative NHEJ. When the engagement of this alternative EJ follows failure of CNHEJ, several of the early CNHEJ factors are present, as already told you, but the process must be abrogated prior to the ligation of the leak 4. And ligation is carried out with either of the remaining ligases, ligase 3 and ligase 1. PARP1 is a sensor for DNA discontinuities, originally shown to operate in base excision and single strand break repair. Previous work have implicated PARP1 in the repair of these alternative EJ. There is even evidence for competition between KU and PARP1 for DSBs, raising the possibility that pre-existing conventional NHEJ factors at the DSB compromise the alternative EJ pathway. DNA end stabilization provided by the conventional NHEJ by KU may be provided in alternative EJ by histone H1. However, it should be emphasized that to date the evidence for a role of histone in alternative EJ is of purely biochemical nature. This alternative EJ is considered to be a mechanistically distinct pathway and it is shown to be active throughout the cell cycle. It is markedly enhanced in the G2 phase as compared to the G1 phase, and is compromised in stationary phase cells tested either in the G1 or G2 phase of the cell cycle. There are speculations that the latter responses may be regulated by phosphorylation of BRCA1 at S988 through CHK2, where in its phosphorylated form BRCA1 promotes error-free NHEJ and suppresses the mutagenic alternative EJ. Therewith it reduces the size of deletions at the breakpoint junctions. However, this dependency is more likely in G2 than in G1 cells, as BRCA1, CTIP and mRN reinitiates DSB resection during SG2 phases, and therefore alternative mechanisms should be explored. There are certain parameters which have positive effects on the alternative EJ, and there are certain others which have negative impacts on them. The positive ones are microhomologies, the presence of ligases 1 and 3, histone H1, PARP1, BRCA1, CTIP, mRN and WRN, and we have discussed about the involvement or presence of these factors in the alternative EJ. There are other factors like the P53 or 53BP1, H2AX, PARP2, KU, DNA ligase, etc. are reported to have negative effects. So, finally, let us once again visit the two main DNA repair pathway, especially for DSBs. We have the canonical or conventional NHEJ repair pathway and the homologous repair pathway. You now know what are the protein molecules involved in taking the decision which pathway it will follow, and you know that KU is an important player in NHEJ, and in HR you have a different set of important critical players, and the formation of certain invading strands takes place in the homologous recombination. In brief, in the homologous enjoining starts with the recognition of the DNA strand by the KU7080 heterodimer, which recreates the DNA PKCs. If the ends are incompatible, nucleases such as Artemis can trim the ends. The XRCC4 DNA ligase 4 XLF ligation complex will seal the break in this process. In homologous recombination, the mRNA CTIP complex starts resection on the breaks to generate single stranded DNA. After resection of the breaks, it can no longer be repaired by NHEJ, and it has to follow the HR pathway. The ssDNA is first coated by RPA, which is subsequently replaced by RAD51 with the help of BRCA2. These RAD51 nucleoprotein filaments mediate strand invasion on the homologous template. Extension of the D loop and the capture of the second end will lead to repair. Thank you.